Did you know how energy is obtained from the food we eat? This can be answered by the electron transport chain. So therefore, this is the electron transport chain, and that is its work. As you hear the name, it transports electrons. Now, where is it exactly? What you saw on the blackboard, you are seeing the outer mitochondrial membrane. This one here is the outer mitochondrial membrane. Mitochondrial membrane. This one you see here is the inner mitochondrial membrane. This one is the full mitochondrion here. Now, this is the outer, this is the inner mitochondrial membrane. Of course, this one here is the intermembrane space. This one here is the intermembrane space. Now, the outer membrane, as we said in the previous video, we said that it has got a lot of protein called purine, and the purine is a transmembrane protein, permitting, allowing passage, allowing crossing of materials from outside to enter or from inside to go outside. Purine, it is made up of a lot of pores. Okay, so materials can enter or leave based on their electrochemical, based on their gradients. Now, this is the electron transport chain. It is made up of, electron transport chain is in the inner membrane. It is in the inner membrane that we have the electron transport chain. It is made up of complex 1, it is made up of complex 2, complex 3, and complex 4. Now, it has got a substance called Q and a substance called C. So that is the electron transport chain. Now, what happens is that complex 1 is reduced NAD dehydrogenase. Reduced NAD dehydrogenase. Dehydrogenase meaning it is good at stealing protons or hydrogen atoms, hydrogen ions from reduced NAD. Complex 2, if you can see it here, complex 2, it's called succinate dehydrogenase. Complex 3 is called, these are complexes of ETC, of electron transport chain. Complex 3 is called cytochrome BC1. That is complex 3. Complex 4 is called cytochrome C oxidase. Cytochrome C oxidase. This is cytochrome BC1. Now, substance Q is called, substance Q is called ubiquinone. Ubiquinone is called ubiquinone. You can even call it CoQ, or coenzyme Q, or simply Q, as an abbreviation with the same, but its full name is ubiquinone. Now, substance C, you see there, it is called cytochrome C. It's called cytochrome C. Now, among all the complexes, it is among all the substances that forms the electron transport chain. It is Q, this Q here, which is a lipid. That's why it is even lipid soluble. Now, another thing I want you to see is that Q is within the hydrophobic domain of the plasma membrane, of the inner mitochondrial membrane. It is within the hydrophobic domain. Now, C, it is on the interface of the inner membrane and the intermembrane space, meaning it is a peripheral protein. It means that C is a peripheral protein. It's a peripheral protein. It's a peripheral protein. Okay? It's a peripheral protein. The other ones are intermembrane proteins. Good. So now what happens is that there is a complex 5, but the complex 5 is not part of the electron transport chain. Candidates get me very well. Complex 5, candidates of senior 6, students of senior 5. Complex 5 is not part of the electron transport chain. Its role is not to transport electrons. It doesn't know about that role. Its role is to transport protons or hydrogen ions from somewhere to somewhere as it is manufacturing for you energy. And therefore, complex 4 is called ATP synthase. Complex 5, is called ATP synthase. ATP synthase. By the name, it means it manufactures for us ATP. Now, the master, have you ever heard of ATP synthetase? Yes, it is also there. When do we call it synthetase? When its role is to consume ATP. As it is doing its work, it uses ATP. I can give you an example of the synthase involved. I can give you an example of the synthetase involved in protein synthesis during activation of amino acids. When a phosphate group is being attached to, when an AMP group is being attached onto an amino acid before transferring A. Now, that one is a synthetase because there the ATP is consumed. But here ATP is formed. If ATP is formed, then the enzyme involved is a synthase, not a synthetase. If ATP is consumed, the enzyme involved is a synthetase. I want you to get used to that language of biochemistry. Now, this is the electron transport chain, complex 1 to complex 4. Complex 5 is ATP synthase. It's not part of the electron transport chain. Now, let's proceed. Now, from the Krebs cycle and glycolysis as well, there is a coenzyme called reduced NAD. This reduced NAD comes from both glycolysis and the Krebs cycle as well. Now we need to get ATP out of reduced NAD. So what happens is that this complex here, reduced NAD dehydrogenase, does what? It oxidizes reduced NAD. In the process, reduced NAD loses 2 electrons. Now these electrons go and enter that complex 1. But at the same time, it loses also protons. Okay? So what happens is that these complexes of electron transport chain are arranged in descending order in terms of electron affinity. What do I mean? For example, complex 1, reduced added dehydrogenase, has got a lower electron affinity than ubiquinone or coenzyme Q. Coenzyme Q has got a lower electron affinity than, for example, complex 2. Complex 2 has got a lower electron affinity than complex 3. So, in terms of electron affinities, it is complex 1 less than complex 2, less than ubiquinone, less than complex 2, less than complex 3, of course, less than complex 4, like that. So in terms of ascending order, in terms of descending order, as you're going the other side, electron affinity keeps on increasing. Now, look, since complex 1 has got a lower electron affinity than ubiquinone or coenzyme Q, then it loses the electron. Remember, it picked 2 electrons from here. Now these electrons, one at a time, is taken to the complex Q, to coenzyme Q, meaning coenzyme Q in the process is reduced. Now, since it is moving from a region where since it is moving from a region from a complex of lower electron affinity to a substance of higher electron affinity, it means that this electron is losing energy, and this energy is free energy. So free energy is coupled to pumping of protons from the it is coupled to pumping of protons from the mitochondrial matrix, because here we have got the mitochondrial matrix here. Here we have the mitochondrial matrix. So protons are pumped from the mitochondrial matrix to the inner membrane, to the intermembrane space. So for 2 moles of electron, which means for 1 mole of an electron, there is a mole of proton pumped to the intermembrane space. There's a mole of protons pumped to the intermembrane space. Okay, that's very important to understand. Now, master, what is in here that picks those whatever? In this complex here, there's what you call iron sulfur centers here. In fact, in complex 1, complex 2, and complex 3, we have got what you call iron sulfur centers. Iron sulfur centers. They are in complex 1, 2, and 3, iron sulfur centers. You all know that iron can exist in various oxidation states. Now, before picking an electron, complex 1, iron is in oxidation state +3. Now, on receiving an electron, iron is taken to +2 oxidation state. Now we shall say that iron is reduced. Now, when iron loses to coenzyme Q, then iron again goes back to +3 after losing the electron to what? After losing the electron to coenzyme Q. So that is the point. Now what happens? The electron continues on this journey. Q goes and reduces complex 3, not complex 2. Follow that pattern very well. So complex 3 is reduced. Now at complex 3, then complex 3 reduces complex 3 reduces what? Reduces cytochrome C. Now, as it is reducing cytochrome C, the electron loses free energy, and this free energy is coupled to pumping of the proton to the intermembrane space. Why pumping? We have got more protons here. So what happens is cytochrome C it reduces complex 4. In other words, cytochrome C is oxidized by complex 4. That's why we call it cytochrome C oxidase. complex 4 cytochrome C oxidase. So complex 4, for it is not going to reduce complex 5. No, because complex 5 is not part of the electron transport chain. So what happens at complex 4, complex 4 is the one which is going to do what? To reduce molecular oxygen. This is the point, this is the only point in the entire respiration where we talk of oxygen that we inhaled. Now, as much oxygen is being reduced, because it is going to do what? It is going to receive 2 moles of electrons to form what? To form an oxide. 2 moles of electrons to form an oxide. Okay, so molecular oxygen is reduced, meaning it is going to receive 2 moles of electrons, then forming what? An oxide. Okay, like that. Now in the process, the electron loses free energy coupled to pumping of protons in the intermembrane space. Now look, I told you that in complex 1, complex 2, complex 3, and complex 4, we complex 1, 2, and 3, we have iron sulfur centers. In complex 4, we have got two domains inside there. We have got iron sulfur, so we have got copper sulfur centers in complex 4. We have got copper sulfur centers, but also we have got a heme group there. Meaning that one is a complex protein. We have got heme group there. So what happens is that copper, for it, on receiving electron from cytochrome C, copper has been in +2 oxidation state. On receiving electron, it is taken to +1 oxidation state. On losing to whatever, to the heme group, then copper loses whatever to the heme group. On losing there, the heme group of course contains iron in oxidation state +3. After losing whatever, what happens is that on receiving whatever there, then iron 3 is converted to iron 2. It is reduced like that. When it is reduced like that, when iron 2 loses to this whatever here, to this oxygen here, what happens is that it is reverted back to complex. It is reverted back to complex +2 +3, like that. So that is the process. That is how it happens. Now these oxides here can react with the protons in the matrix to form what? Protons in the matrix to form what? To form water. We can form metabolic water there. How can we balance? We can balance by putting a 2 here. Now our equation is already happy, is already balanced. We are forming molecular water there. So what happens is that now, master, you have not yet hinted on complex 2. I'm coming, cut the pressure. So what happens? This reduced NAD, on losing electrons, it was oxidized to oxidized NAD. In other words, from reduced nicotinamide adenine dinucleotide to oxidized nicotinamide adenine dinucleotide. So what happens? It was oxidized to oxidized NAD. Now look, complex 2, it is called succinated hydrogenase. Now, complex 2 is the only complex that oxidizes. It is the only complex. Complex 2 is the only complex capable of oxidizing reduced flavin adenine dinucleotide, okay, to oxidized FAD, to oxidize flavin adenine dinucleotide. Now, complex whatever, that is where we find the only link between Crawford cycle and electron transport chain. So as FAD is being oxidized, electrons are being removed, and these electrons here are shuttled to Q, to coenzyme Q. Coenzyme Q picks them. Then this coenzyme, now the energy, they are going to lose free energy. But now, no pumping of protons into the intermembrane space. Why? This complex here, complex 2, is not a transmembrane protein. It ends somewhere. It is just integrated into whatever, but it does not span the entire whatever. So what happens? It does not what? It does not pump the proton into the intermembrane space. But now, As this electron from complex 2 is heading towards reaches complex 3, then to cytochrome C, the free energy it loses is used by complex 3 to pump the protons into the intermembrane space. In other words, this complex 3 is very important for the functionality of complex 2 because energy from its electron it loses for free energy. It is complex 3 that uses it to pump protons, whatever. Now, consequently, oxidation of reduced NAD results into pumping of 3 per electron, pumping of 3 protons into the intermembrane space. Oxidation of reduced FAD results into pumping of 2 protons into the intermembrane space. Like that. Consequently, there is a buildup of protons into the intermembrane space. There is a buildup of protons into the intermembrane space. A buildup of protons into the intermembrane space. There is a buildup of protons into the intermembrane space. A lot of them are going to concentrate inside here into the intermembrane space. So now, since these protons are taken from the matrix into the intermembrane space, there exists a gradient called electrochemical gradient. Electrochemical gradient develops between the matrix and the intermembrane space as far as proton concentration is concerned. Now, electron because of difference in charge, more positive charge to the intermembrane space than to the matrix. Secondly, chemical, more protons into the intermembrane space than in the matrix. Now, because of that, because of the electrochemical gradient, protons intend to diffuse back. But as we said during the discussion of actual structure of mitochondrion, the inner membrane is restrictive in nature. You don't just pass anywhere as whatever. So what happens? They can only go through the channel of ATP synthase. These are the bulbous heads you see here, the stocked particles. Those stocked particles, I see them, stocked particles on the crystal. Now, this is ATP synthase. Now, ATP synthase, what happens is that with ATP synthase, what happens is that it has got 2 domains. It has got the F1, F0, F0, then it has got the F1. So F0 is part of that enzyme ATP synthase embedded into the inner membrane of the mitochondrion. F1 is the domain that is outside, having this bulbous head here, the stocked particle here. Now, this F1 here is the one which is going to form for us ATP. Now, protons are going out to diffuse back into the matrix. What is inviting them as they are returning? What is inviting them? So, what is inviting them to return? What invites them is the electrochemical gradient. So as they are returning, they go through. This head is having very many domains, epsilon, what, gamma. So as it is returning, the chemical energy, the kinetic energy from the diffusing protons is now converted to the chemical energy from the diffusing protons, or kinetic energy, as they are diffusing, is now taken up by the stocked particle, by the F1 domain of the enzyme ATP synthase. Now this bulbous head starts to rotate. It rotates, okay? As it is rotating, as it is moving, then the energy from that rotation is coupled to combination of ADP plus an inorganic phosphate to form for you ATP. And that is how we have managed to form our ATP from the reduced coenzymes, okay? From the reduced coenzymes. So that is what you call the electron transport chain. So these protons you see here, they are the ones which are going to react with water, with the oxides, to form metabolic water. So that is the electron transport chain, and that is how ATP is made from the food we eat. So that is very important, and this ATP is going to drive a lot of whatever formation of sperm cell movement, for example, movement of some cells like sperm cells, for example, muscle contraction, transmission of electrical impulses, or whatever, contraction of muscles, very many whatever active transport of materials during synthase, for example, during the activation of amino acid during protein synthesis and other whatever. So that is very, very useful energy. Feel free to ask, where do you think I have not explained very well, but also appreciate the part you have understood from this video? What is the thing you think I need to clarify more on it? Feel free to put in your comments in the comment section as you follow my YouTube channel, Biology King Yugi. Because on TikTok, I put there just part of the video. Full videos on YouTube. You know that the algorithm of TikTok sometimes does not easily push long videos. So I put there part of it, but the full video is on Biology King Yugi. You just go there, then you go to the playlists. Now there's a playlist called the Senior 5 Respiration. That's where you're going to find a long video for this whatever here. Feel free indeed to ask me, where do you think we need to improve? But also appreciate what you have understood from this video. Contribute and whatever. You also, my fellow teachers, you can come up and try to contribute on the video. You can try to clarify somewhere in the comment section. I'll be very happy. Let's work together to promote, to make sure that our learners are not defeated by biology. Thank you very much.